All right, so good morning, everyone. It's a great pleasure and joy to introduce you briefly to our speaker today, Professor Francis Chan. Francis is one of the leading scientists in the field of innate immunity, cell death, particularly emphasis in necroptosis these days. And he agreed to speak from faraway land from us, China. So it's evening for Francis now. It's very early for us here. Francis moved recently to China from the Duke University in the US. He's now the professor of immunology of Zhejiang University. And I think I briefly mentioned, but now we are trying to establish a collaboration between China and Brazil in immunology community. So we've been also attended to seminar series that Francis is organizing for us in every Friday or every other Friday or some of the Fridays morning. So Francis, thank you so much for being here with us. It's a real pleasure and hope to be able to get you here in Brazil very soon.
Well, thank you, Gustavo. So let me see if I can share screen. Can I share screen? So here, where'd I go to share screen?
Okay, Marcio, is it?
I think I need to be a co-host or something.
Márcio, você coloca ele como co-host para ele conseguir compartilhar a tela.
Ele já tá.
Okay, so we already have Francis. Try again.
I already have it. Okay, all right. So let's see. How come that I still don't see that?
All right.
Just give me a sec. It's been a while since I used Zoom because I here they use something different, but I'm still not seeing that icon that allow me
So
to.
in the bottom you don't see share screen, or you don't see in the share screen area you don't see.
I don't see share screen for some reason.
It's in the bottom. If you move your pointer to
yeah, there you go. There you go. Now it's active. Okay.
Okay.
Let me see. Okay. Share. Okay. First, I need to allow the privilege. Just give me a sec.
No, it's fine. No worries.
Okay. I think I now can do that. Okay.
Yes.
All right.
Perfect.
Can you see my screen? Perfect.
Yeah.
Okay. Let me put it on.
Presentation.
Okay. Well, thank you. First of all, to Gustavo, Karina, and everyone, my Brazilian friends. I kept telling people, after I landed, in China late last year, that I have many friends in Brazil, and they were shocked, and actually, right, as Gustavo said, we are starting, this online seminar in a series where scientists from both sides. We just introduce what we do, and hopefully that will lead to collaboration down the road in the future. And when I told people why I wanted to do this, they was actually quite surprised that I even have or know anybody in Brazil, and in fact I mean that's the case, right? I knew a lot of you in Brazil, and I visited Brazil several times, and every visit was a real treasure. And so again, thank you for having me here today. I know it's very early for all of you. I'm gonna try to make it sort of fun as much as possible, and for the students in the audience, please feel free to interrupt me at any point if you have any questions. Okay. So as Gustavo said, I have been in China for the last couple of months, and just to give you like a glimpse of what the city looks like. So this is a picture.
gosh.
Yeah,
Beautiful.
this is the famous West Lake. And so it is a city, where I'm situated, a city called Hangzhou, it's very near Shanghai. And it's a city, really steep, in history, and culture. And so I would love, to have you guys, come visit, and maybe, not just PI, but even students, in the future, if we can actually have students going back and forth, as part of the exchange experience, I think that would be wonderful. Okay, so with that, this is the sort of like outline and lecture today. Today we're going to talk about necroptosis. The title that Gustavo gave me is RIPK3 necroptosis, and antiviral responses. So this is, believe it or not, right? It sounds like a pretty sort of like focused topic, but there's really quite a lot to talk about. So we're going to try to cover all these sort of like subject areas today in the limited time that we have. So now I think for the last couple of days you already have heard from other speakers in this seminar series about different types of cell death. And of course, when we think about cell death, the first form of cell death that we know of is apoptosis. And so it is a quiet way, for the cell to exit the stage, so to speak. The word apoptosis. You can see. Let me see if I can bring out a pointer. Let's see, pointer. Okay. Can you see my cursor? For some reason, the pointer is not showing.
Yes, we can see it.
Okay, you can see it. Okay.
Yeah.
All right, good. So I'll just use my pointer, my cursor, my mouse. So the word comes from a Greek term that means falling leaves. So this again, I think is a familiar to all of you. So apoptosis is really most well known for its role in sculpting our body parts during development. And but of course, there are situations where you will also go for apoptosis as a way to eliminate injured cells or like damaged cells. So I always like to sort of like tell people like for immunologists, right? Apoptosis is kind of like a very clean way for you to get rid of sort of like that cell debris from our body. It's kind of like you have a marked trash can, right? Because the apoptotic cell will flag itself for phagocytosis on the cell surface with exposure of phosphorylated, and that allows phagocytes to come in and clear them out. So it's just a very clean way to get rid of the cells without inducing inflammation. Now, you contrast that now, nowadays, of course, we know, that beyond just apoptosis, there are many different types of programmed cell death modules. So this is taking out from, again, a review, that many people, in the audience might actually have contributed to a few years ago, and you can see, in this slide, spinning wheel, so to speak, many different forms of, cell death, including, the subject, of today's discussion, necroptosis. And so, what is necroptosis? Now, so again, I think for cell death, we always like to fall back on morphology as a gold standard to define what is specifically sort of like lytic cell death versus apoptosis. So here is an electron micrograph showing you a T cell that undergo necroptosis, and you can see a very sort of like significant feature of necroptosis is the sort of like extensive disintegration of the plasma membrane. And in doing so, of course, right, the cellular content leaked out, and it, it's kind of like, a volcano eruption. And the ashes, and smoke that came out, from this necroptotic cell, or what we refer as immunologists as damage-associated molecular patterns, are very potent inducer of inflammation. So it, unlike, apoptosis, therefore, can trigger, a series of immune reaction that culminate, in sort of like, inflammatory diseases. So that's the paradigm, right? And that paradigm, for at least in the case of necroptosis, really come from studies, that I highlighted, in this slide here. So, we'll start off, with, the study at the top here. So this, is a situation where, if you were to sort of like, in mouse, inactivate, an essential apoptosis adapter molecule called FAT in just the skin epithelial tissues, then you get a very aggressive and spontaneous inflammatory reaction. Now, that spontaneous inflammatory reaction is really driven by excessive necroptosis, because you can actually reverse that and restore normal skin tissue structure by inactivating at the same time the essential molecule for necroptosis RIPK3. And you can also see a similar situation for intestinal epithelium. So you knock out, again, FAT or, the caspase molecule that it recruits, caspase A, you get the same results. So here, we're looking at the sort of like, immunohistochemistry staining for a lysosome, marker. So this is marking, pen cells. You knock out, FAT and therefore inactivate apoptosis, and you get excessive necroptosis and tissue injury in the intestine, which again can be rescued, if you inactivate also at the same time, RIPK3. If you extend that, to beyond just these epithelial tissues and look at the whole animal, you see the same situation as well. So here, we're looking at not just FAT now, we're looking at caspase A. And so if you knock out caspase A, this is the caspase A knockout embryo here. During development, you can't even get a normal embryo developing. You can see in this picture here, the sort of like rapidly reabsorption of the dying embryo. Now, if you however inactivate RIPK3, then you can restore normal development of the embryo. So that indicates that again, just like what I was saying a moment ago, necroptosis really can cause very detrimental inflammation. So that, because of all these data that I just went through with you, has been the paradigm in the field for a long time. Now, so however, when we talk about necrosis or necroptosis, really, you can actually date back way beyond the recent sort of like 10, 20 years. So, the earliest, sort of like, recorded, sort of like, information on sort of like necrosis at that time, and of course, the term necroptosis was really only coined more recently. But, back, in the turn of the 20th century, physician, scientist, in New York City, by the name of William Cody, so he was seeing a lot of like, cancer patients, right? They walk into his clinic, and somehow he found a way, to develop, a concoction of bacterial extract. And he realized that, when applied on these patients, right, this concoction of bacterial extracts actually managed to actually cause regression in many of these tumor patients. And so because of that, it got his name on the front page of New York Times. At that time, you can see, right here. Now, of course, at that time, that was, sort of like, quite exciting. And then people even coined the term, Codis toxin, to refer to this, so mysterious, concoction of bacterial extract, but then the active ingredient wasn't known until the 1970s, when Lloyd Ode discovered that, the active ingredient inside of Codis toxin turns out to be the cytokine that we're going to be referring to quite a bit today, tumor necrosis factor, or TNF. So a little bit of a history, of course. There's been many landmark studies in the necroptosis field over the years, and we will cover many of them, but because of time, we won't be able to cover all of them. But I just want to highlight a couple of them, maybe more like, to me, in my mind, to be sort of like pretty important discoveries along the way, right? So the first, like after the discovery of TNF, I think the next sort of like important discovery was made in 1988 by Linda Gooding, who since has passed away. And so she discovered that in L cells. So nowadays in the laboratory, if we want to study necroptosis, we oftentimes use this mouse cell line called L929 cells. So that's the L cells that Linda Gooding was using. And so what she observed was that when you apply TNF to this mouse fibrosarcoma cell line. The cells can actually undergo more than one form of cell death. She was able to actually observe two different cell death morphology, one that resembles apoptosis and the other one that looks like necrosis. So that was that. And then, of course, now you fast forward 10 years later, Walter Veers and Peter van den Beele, pictures shown here. They discovered that in L cells, if you were to inactivate caspases, and at that time they don't know exactly which caspase is responsible for this phenomena, but then when you inactivate caspases, you can actually further increase this necrotic cell death response. And again, just like what I was showing you, a moment ago, with the genetic, experiments where you inactivate either caspase A or the adaptive molecule for caspase A fat, you will get, sort of like exacerbation of necrosis. And so, like, Peter, and also Walter Veers, they made that observation, as early as like, in the late, 1990s. And so that, again, it will be something important that we will come back to later on, right? Which is that inactivation of caspases is a key priming signal for necroptosis or necrosis. And then, you move forward, to the year 2000, the Yook Sharp, the late Yook Sharp, here, he, published a paper in Nature Immunology describing, essential role for RIPK1. And in fact, at that time, they don't even call it RIPK1 because, the other RIP, kinases, many of them has not been cloned, so it was simply referred to as RIP, to be an essential, adaptive molecule for necrosis driven by TNF, Fas ligand, and also trail. But of course, then, the field sort of like got stuck there for a long time, in part because it was very difficult to study biology of this form of necrosis because the only sort of like downstream signal adaptive molecule that we know of that is essential is RIPK1. And of course, we now know that, if you knock out RIPK1, for example, in the germ line, the amino also failed to develop normally. So that tells us that, it is essential, for survival. And because of this requirement for survival, and you don't have an animal, viable animal model to study the physiological function of necrosis or necroptosis, it just becomes very difficult to push the field forward. Now that changed a little bit because in 2005 and 2008, Junyin Huang published two papers. The first one, discovering from a chemical compound screen, a compound that we now know as necrostactin-1. And since then, she has also developed, and other people have also developed, other derivatives, RIPK1 inhibitors. And so this is an inhibitor, and the first chemical inhibitor that can block necrosis or necroptosis driven by TNF. And so in 2008, then her group further elucidated the target of necrostactin-1, and that turned out to be RIPK1. So now you have a drug that you can use and apply, in theory on animal, and therefore, it's sort of opened up, a window of opportunity to study the biology, of the pathway in physiology. But of course, that is still a drug, right? And necrostactin-1, for those of you who have actually used it, is actually very biologically unstable. But that changed, right? In 2009, when 3 papers, published back to back, and back, by Sheldon Wan, Joy Huan, and myself, we found RIPK3 to be essential kinase that promote necroptosis. And because RIPK3 knockout animal, they are viable. So I think that really does open up the opportunity and possibility for further research. And in 2012, then Sheldon Wan and Zhang Ganyu, then identify the effector molecule, mixed lineage kinase domain like or MLKL as the key effector for necroptosis. So these are the sort of what I would consider to be pretty essential discovery in the field of necroptosis. Now, so let's just come back a little bit and then talk about the pathway components and how they're regulated, right? So if you activate, and now we know that classical necroptosis are activated by TNF, and the receptor responsible for the response is TNF receptor 1. And so the first molecule that is activated is the kinase RIPK1. So RIPK1's got several parts that are essential for this function. The first one being at the N terminus, the kinase domain. And then RIPK1 has in the middle of the protein a portion, that is referred to as the RIM motif or RIP homotypic interaction motif. We'll come back to that, later on, and to tell you more about, what this is. And then beyond the RIM motif, which is a protein interaction motif, there's a third domain in the C terminus called the DEF domain. So the DEF domain is the portion that allows RIPK1 to be anchored on the receptor signaling complex. Now, so RIPK1, because of its important function in cell death and also inflammation, it is heavily regulated by different types of post-translational modification, including phosphorylation and ubiquitination, etc. Now, this will not, however, be the focus of our discussion today, because we're going to talk more about the biology instead. But, under certain conditions, however, RIPK1 can actually engage and recruit RIPK3 through the RIM motif. But however, normally, when I refer to normally, by that I mean in a cell that has active and normal caspase A function. This reaction is actually unstable. And the reason being that caspase A can actually cleave among its substrates, RIPK1 and RIPK3. And therefore, the cleavage will basically stop the necroptosis reaction right on its track. And because of that, we now will the field rightly recognize that caspase A inhibition is a key priming signal for necroptosis induction. And you can do that by like what I showed you a moment ago, genetic inactivation of either caspase A or its adaptor molecule FAT, or you can use pharmacological inhibitors such as caspase inhibitors. And once you met that condition, you inhibit caspase A, then you can have stable interaction between RIPK1 and RIPK3. RIPK3 become phosphorylated, and they will go on and phosphorylate its downstream effector molecule, MLKL. Now, MLKL has two portions in its protein, one in the pseudokinase domain, and the other one, the business end, the 4-alpha helical bundle. RIPK3, when it's activated, will phosphorylate, MLKL in the pseudokinase domain. And this phosphorylation event will allow it to undergo a organization and translocate into the membrane to form supposedly pores. And this is how you get the membrane rupture that I showed you earlier during necroptosis. Now, we know that in recent years, from research from many different groups, that RIPK1 is not the only way that you can induce necroptosis. You can also induce necroptosis independent of RIPK1 through two different types of upstream adapter molecule. The first one is the total receptor 3 and 4 adapter, TRIF. Now, TRIF, just like RIPK1, also engage RIPK3 via the RIM and RIM motif interaction. And there's a third pathway that you can activate RIPK3 and MLKL, and that is through yet another RIM domain containing adapter molecule called CBP1. And this pathway oftentimes is activated during virus infection. We'll come back to that later on. Now, no matter what the upstream activating signal are, the downstream events that culminate in activation of RIPK3 and MLKL are believed to be the same regardless. And it is interesting, however, also to note that in all three cases, whether you're going through RIPK1, CBP1, or TRIF, in all three cases, these molecules, they have all been shown by different groups to have functions in not just cell death, but also in innate immune response. So I think that right away, from a molecular architecture perspective, tells you that necroptosis likely has a pretty important role in controlling inflammation. All right. Okay. So now, you know that inhibition of caspase A is a requirement for necroptosis induction. So the question then really is, when does it actually happen in pathophysiology? So for that, early on, what we think about is potentially, in certain types of virus infections. So this diagram, taken out from a review article written, in the early 2000s, shows you, all the different ways a virus can actually manipulate, the host immune response. And among those, viral strategies that can manipulate and modulate the host response, strategies that target caspases, shown in this red box here. So because I just told you already that inhibition of caspase A in particular is a priming signal for necroptosis. So that led us to think that perhaps we can actually look at that as a way to sort of like illustrate whether necroptosis can indeed happen during normal physiology. So the idea once again is that many large viruses, and by that I mostly refer to pox virus and also herpes virus. They are known to encode caspase inhibitors, and therefore you can envision a situation where the infected cells will become susceptible to necroptosis because you met the criteria for sensitization to necroptosis. And so let's use vaccinia virus, for example, to highlight how necroptosis pathway actually participate in antiviral immunity. So just a little bit about what vaccinia virus is, right? So it's a large virus, and it's an envelope virus, and this is a structure here. And what's interesting about vaccinia virus, so there's a genome architecture here. So this is at the very two ends of the viral genome are two regions that are referred to as the inverted terminal repeats. Now, these two regions are really interesting because this is a region that is actually highly variable among different species of the pox virus within the pox virus family. And this is a region that encodes many immune modulatory genes as well, and including the caspase inhibitor that I will be talking about. So early on, right, in terms of vaccinia virus and its relationship to necroptosis, as early as in the 1990s, we as in the field has already understood that if we were to actually express TNF within the virus and therefore produce a local concentration of TNF, you can actually very effectively control the viral replication. That tells us that TNF at least is important. But then the data is really much more sort of like solid and relevant when you look at later on in this paper that we published in 2003. When you look at TNF receptor knockout animal, you can see in black here, these are the knockout animal. If you were to infect wild type in open circle here and the knockout animal lacking TNF receptor, you can see without TNF receptor, and you can do that for TNF receptor 1 or TNF receptor 2, you get the same results. You will get a exacerbated viral replication and viral load. So that indicates that TNF indeed is important. So again, I mentioned earlier that TNF is a classic cytokine that can drive necroptosis research, so that becomes relevant. Okay, so now what's next? Okay, so we also in the field discover in the around year 2000 that TNF if you add TNF to vaccinia virus infected cell, you can drive cell death, and not only that, it because as I told you a moment ago, we already know that vaccinia virus encode these caspase inhibitor. So the caspase inhibitor in vaccinia virus is a molecule called B13R. You can actually delete that from the virus, and if you take this mutant virus that can no longer inhibit caspase, you use that to infect cells, and you ask whether the cells can now undergo cell death in response to TNF. You realize that now the infected cells is no longer sensitive to TNF-induced cell death. So this experiment tells us that not only is the virus encoding an inhibitor for caspase, but this inhibitor is actually important in conferring sensitivity to TNF-induced cell death. Okay, so all of that, right, is a long way to lead us to this model that we propose. That is that when you have the host cell, if it becomes infected by vaccinia virus, a lot of things of course happens, and among these is that the virus will inhibit caspase activation. And we also know from data that I just showed you a moment ago, TNF is going to be produced very early on during the infection, and because the virus can inhibit caspase A, maybe, this inhibition will shunt the response, from apoptosis driven by TNF to one that causes necroptosis. Okay, so, and indeed, that turned out to be the case, right? So shown in this little diagram here, if you just focus on the graph on the right, you will see that here we're doing an in vitro tissue culture infection in a cell line that lacks RIPK1. And you can see that in the virus infected cell, unlike the uninfected cell where in adding TNF the cells will undergo cell death. But then in the virus infected cell now, without RIPK1, it can no longer undergo cell death. But in a cell that contains a wild type copy of RIPK1, the infected cell will undergo cell death in response to TNF. So this data tells us that not only is TNF inducing cell death in the virus infected cell, but this cell death actually does require RIPK1. So with that, when we discover RIPK3 to be the essential kinase in the necroptosis pathway, we of course then wanted to know whether RIPK3 is similarly required for TNF-induced cell death in virus-infected cell. And you can see in this graph on the left here that infected cells, in the wild type situation, they can undergo cell death just like what I showed you a moment ago. But if the cells actually lack RIPK3, now this infected cell become resistant to TNF-induced cell death. You can also look at the sort of biochemical hallmark of necroptosis, which is the association of a complex between RIPK1 and RIPK3. Now, normally in an uninfected cell, you need TNF and CVAD, right, blocking caspase in order to see the complex forming. But then in virus-infected cell, all you need is now TNF. You no longer need to block caspase with the CVAD inhibitor, the reason being that the virus now provides this inhibition in the form of the caspase inhibitor that it encodes. And now with TNF alone, you see the complex form. So that was that, okay? But of course, it's a tissue culture, right? How do we know that it's actually physiologically relevant? So we did an infection in mouse, and the first thing we wanted to know is, is TNF actually produced? So sure enough, right, within a day of infection, you can see immune cells are beginning to produce TNF. Not only, are you seeing the TNF production, but if you look at actually tissue extract here, we're looking at the liver extract, you can see a transient induction of the complex between RIPK1 and RIPK3 between 12 and 24 hours. So there's TNF production, there's the molecular signature of necroptosis. And when you look at the tissue, morphology, you can also see that in the wild type situation, there's a lot of inflammation, and all the inflammatory cells are focused and localized in regions where you have necrotic tissue. Now, if you do the same infection now, however, in an animal that lacks RIPK3, you do not get necrosis in the tissue. You also do not get inflammation. So there's a clear correlation between the two. But then what is the consequence for not having necroptosis induction in response to this virus infection? The consequence is actually quite dire. You can see on the graphs at the top here, we're looking at viral load in different tissues within 3 days post-infection. And this is on log scale, so you can appreciate that within 3 days of infection, if you do not have RIPK3 and therefore you cannot induce necroptosis, there's anywhere from 100-fold increase to over 1,000-fold increase in viral load in the animal. And the consequence of failing to control the viral replication is that the animal will eventually die because of the infection. So that was that, right? So we used vaccine virus early on to show that like the pathway of necroptosis is really physiologically relevant. But you might be wondering, right, what is vaccine virus? Well, no, suffice it to say, right, we now, we've lived through several years of the pandemic, right, with COVID-19. So we all know, what vaccine is. The word vaccine comes from vaccinia virus. So the vaccine virus is actually a vaccine strain that we use to vaccinate the world population against smallpox. And so it's a highly effective vaccine, and that's the reason why we stick with the term vaccine, right? Use the word vaccine virus to coin the term vaccine. And so Yerner, early on, however, was using cowpox to vaccinate. So this is the sort of earliest history in record of sort of like, early vaccinology, so to speak, right? So what is vaccinia virus? So vaccinia virus, of course, unlike smallpox, it's a live virus infection, it's a live vaccine. It causes, however, a very benign infection in the host. And so this is what you want, right? For a live vaccine, you don't want to cause severe disease. Now, it is, however, at the same time, still retain a very highly immunogenic, property. And therefore, of course, you want that, in a vaccine, right? But what is interesting about vaccine virus is, back in the days when it was actually found that vaccine virus has these properties and therefore they're perfect, to be a vaccine strain, they actually do not know what is the origin, where it came from. There's actually no known natural reservoir, so to speak, for vaccine virus, and that's for sort of like a conversation and discussion on a different day, perhaps. So when we actually, think a little bit more about why we ended up using vaccine virus, and we do a genome comparison. So this is basically a genome sequence comparison between vaccine virus and some of the other more naturally found pox viruses. So all the color boxes here that I'm showing you in red and yellow and so on and so forth, right? They represent genes that are either missing entirely or severely truncated or fragmented in vaccine virus, but are nonetheless found in the naturally found pox viruses. So all these are very interesting and makes you sort of wonder. And remember, I told you earlier that these genes, right? They were all basically concentrated in the two ends of the viral genome. And if you recall, I said what I said earlier. These two ends are what we refer to as the inverted repeats. And this is where the virus encodes a lot of these immune modulatory genes. So it really begs the question, right? These missing genes in vaccine virus, could they actually, be encoding something that can actually interfere with host response, and in this particular case, host cell death by necroptosis? So that we want to look at that, and we perform this experiment here. Where we take mouse embryonic fibroblasts and we infect them with recombinant virus that encodes GFP. And so you can track infected cells with GFP on the x-axis here. And y-axis here, we track cell death with PI. And so you can see with TNF, right? If you look at, for example, uninfected cells, there's no cell death. But then in vaccine virus infected cells, just like what we have already known for many years, they become sensitive to TNF-induced necroptosis. There's a reduction in GFP and also an increase in PI signal. But in cowpox virus infected cell, you get completely the opposite result. There's no cell death whatsoever. So this is really intriguing. And then we wanted to know what is going on. To cut a long story short, right? So just to remind you again, in terms of TNF-induced necroptosis, these are the three essential molecules we're talking about: RIPK1, RIPK3, and MLKL. So when we just do a very simple Western blot, this is what we saw. And the result really jumped out from the page, right? So cowpox-infected cells, we have almost a complete disappearance of the RIPK3 protein expression. So that was quite striking, and you don't see that in vaccine virus infected cells, for example. So the virus is really targeting RIPK3. And to, again, cut a long story short, we, through using, different types of approaches, including using protease inhibitors, we're able to see, and demonstrate that the only type of inhibitor that can rescue this disappearance of RIPK3 protein from cowpox infected cells are proteasome inhibitors. So that means the virus cowpox is causing the disappearance of RIPK3 protein through a proteasome degradation pathway. So we went on and did an siRNA screen, and we found in cowpox a viral protein that encodes a structure that looks like this, with a series of anchoring repeats at the N terminus and an F box at the C terminus. Now, it's because of the functional consequence that I'm going to tell you more later on, we now refer to this viral protein as viral inducer of RIPK3 degradation, or VRD for short. Now, you will also recall, right, because we started out seeing that cowpox infected cells, they were resistant to necroptosis, but then vaccine virus infected cells, they are sensitive. So by definition, if this protein VRD is what is responsible for the disappearance of RIPK3 that I showed you a moment ago, by definition, this gene should be either mutated or absent or changed in vaccine virus. And that turned out to be the case. So here I'm only showing you two examples of two different strains of vaccine virus that has a genome sequence. And in both cases, they encode a truncated version of VRD. Now, if you look at other strains of vaccine virus, in some cases, they don't even have this truncated version entirely. So this again really just satisfy another very important criteria for VRD, and that is that it has to be missing, or at least the function should be missing or lacking in vaccinated virus. So now I just show you, in the cartoon, earlier, what this VRD protein looks like. It has a F box. So one might be wondering, what is an F box? So it turns out, in the mammalian genome, we encode many different types of F box proteins, and they all are basically adapters, for this cellular machinery that we refer to as the SCIP1 cotton and F box complex, or SCF complex for short. So this complex is basically a E3 ligase complex. And so the F box within the complex, its function is kind of like the fishing hook. It is to recruit basically substrates for ubiquitination, and then further on going on to the proteasome degradation pathway. Because VRD also has an F box, so it's actually quite straightforward to propose this model where when the virus infects the cells, VRD expression will basically co-opt and displace the endogenous F box, and allowing this SCF complex now to redirect its specificity towards RIPK3 for ubiquitination and further on degradation. So is that the case? So to obtain evidence, we generated a virus, a mutant virus that lacked VRD, and you can see now, compared with the wild type virus, it no longer can degrade RIPK3. You can also use siRNA, to knock out the expression of the cellular components of the SCF complex. In this case, I'm showing the results for column 1. You knock it out, now the virus also cannot degrade RIPK3. And the functional consequence of that is, whereas wild type cowpox, just like what I showed you a moment ago, the cells are resistant to necroptosis. But if you now remove and inactivate VRD from the virus, the infected cells now again regain sensitivity to TNF-induced necroptosis. So what I've shown you so far is basically two examples, right? So they're both pox viruses. Vaccinia doesn't, encode a VRD, and the infected cells become sensitive to necroptosis, whereas cowpox encode a VRD and the cells are resistant. So if you look at sort of like the whole family, what do we see? Do we see and what turns out to be the case is we see when we look at the whole orthopox virus family, in almost every single case, the virus encode an intact copy of VRD. And we can show also in some cases that these VRDs from other pox viruses, they can also degrade RIPK3. So really, what that tells us is, we got very lucky, early on by using vaccine virus as our first pathogen, because vaccine virus is the exception to the rule. And if we were to use other pox viruses, we would not have seen the virus infected cell become sensitized to necroptosis, and it would have taken us a lot longer to actually figure out the biology for the cell death pathway. Now, so all of that again is just biochemistry, right? So in the model that I showed you a moment ago, which I'm showing you once again here with a little more details, you can also see that the infection and expression of VRD has two separate consequences. One, which I focused on earlier, is this redirection of the substrate towards RIPK3. That we understand, and I showed the results for that already. But another consequence for VRD is it will displace the endogenous F-box proteins, and therefore, in doing so, alter the turnover of the endogenous F-box protein substrates. And one of these substrates turned out to be I-kappa B alpha. And of course, we know I-kappa B alpha is a regulator for NF-kappa B activation. And when you talk about infection with a virus, of course, NF-kappa B is going to be very, very important, right? It turns on all kinds of like cytokines, chemokines, and whatnot. So it could well be, and in fact, when we look at, the virus-infected cell in tissue culture, indeed, the degradation of I-kappa B is severely, impaired. So that, really, just, raise a very important question, and that is that, in physiological infection in a whole organism, which one is going to be more important, right? Is VRD actually sort of like mediating, whatever, its pathological effects, through targeting RIPK3 or targeting, for example, NF-kappa B? So to answer this question, we did the following experiment here. So we take, wild-type virus or the mutant virus that we like, VRD, and in fact, mice, that are actually, wild-type or are lacking RIPK3. You can see that when you take out VRD, the virus is actually not as fit, and therefore it doesn't replicate as well in the host. Now, however, if this effect is entirely driven through RIPK3, you can imagine that taking this virus now, doing the same infection now in a RIPK3 deficient animal, you should now restore normal replication of the virus, right? And indeed, that's the case. So I think this data tells us that although VRD can also at the same time interfere with NF-kappa B activation, the physiological relevant target for VRD really is RIPK3, not NF-kappa B. Now, so we went on and looked at what are the other consequences for VRD. So we look at, for example, in this case, inflammatory gene expression, and we see a very clearly distinct pattern based on whether the virus has VRD or not. But what is quite striking is when we look at, what these changes are, then we see that, for example, right, because VRD can target RIPK3 degradation, and if you recall, once again, the dogma in the field is necroptosis drives inflammation. So we expect that in a wild-type virus infection, because the virus can degrade RIPK3, you get less inflammation, right? That's not the case. So here, you can look at, for example, at the top here, this is the histology, a lot of inflammation in wild-type cowpox infection. You take out VRD and therefore retain wild-type RIPK3 function. You actually get reduced inflammation. You can see that also by flow cytometry, looking at neutrophil infiltration into the tissue. Also, once again, you retain RIPK3 by inactivating VRD, you get less neutrophil infiltration. So that's polar opposite to what every review that you read tells you about, right? You can also look at specific chemokines, and that tells the same story. So you don't have VRD, and therefore you retain wild-type RIPK3 function. You actually get less chemokine expression. So that's counterintuitive. And I think, I just want to use that to highlight, something, right, important here. And that is that, what we read, perhaps in review, we need to take it with a pinch of salt, right? So in certain situations, having necroptosis may not mean like, having, more inflammation, so to speak. Now, so I'm going to also want to emphasize one other thing. So here, in this diagram, as I told you a moment ago, right? So vaccine virus is really, the exception to the rule. It doesn't have VRD and it causes a benign infection in its host. All the other cases that we look at, and I'm only listing a few of them, monkeypox, again, so last year, there were also, some reports, around the world, of outbreaks of monkeypox. So we know, because of these reports, that even monkeypox in human infection is pretty nasty, okay? And of course, smallpox is really, really nasty. So it almost looks as if, right, that when the virus encodes VRD, then it increases, the pathogenicity of the pathogen, right? And then when you remove it, then you make, the virus more tame. So it looks like, there's a pretty interesting correlation. So we're really excited about this hypothesis until we ran into this, other virus, which is myxoma. So what is myxoma virus? So myxoma virus does not have VRD. Now it infects rabbits. Rabbits is its natural host and reservoir, and it causes in rabbit a very deadly disease. So here's one example to highlight and illustrate what I meant when I say okay, the virus causes a very severe disease in rabbits. So in Australia years ago, the rabbits were running wild, and they were really breeding and just the population just completely went berserk and out of control, to the point that they were running wild. They were basically just like feeding and grazing on the crops, and it's causing severe economic damage to the local farmers. So how do they actually deal with this wild rabbit problem? Is to introduce myxoma virus into the wild rabbit population. And of course, sure enough, because the virus is very deadly to rabbits, it was actually very effective control of the wild rabbit population. So this is all just sort of like interesting anecdote to illustrate that this is a deadly virus, but nonetheless doesn't have VRD. So are we wrong when we propose a model that VRD might actually be sort of like driving pathogenesis of the pox virus? Now, so the answer turned out to be actually already something that we discovered a few years ago in a review article that we wrote. This is really no secret. We're not the only ones who have observed who's made this observation. So it is actually quite widely known in the field that in many mammalian species, we actually found examples that they do not have a functional RIPK3 gene. But then the relevance or meaning for why certain species do not encode a functional RIPK3 gene was unknown. But then what we realized is that rabbits turn out to be one of these species. So you can almost make a very interesting argument, right? Here, that if you take the RIPK3 gene away, and therefore now the animal, the rabbits, no longer can actually trigger necroptosis. And if you buy the argument that necroptosis is an antiviral response, then the pathogen also now no longer has to carry this burden of carrying a countermeasure against this host immune response. So I think it actually is a very interesting idea. Of course, it's difficult to actually prove that, but I think it's nonetheless something interesting to consider. Now, so what I've told you so far is that in vaccine virus, right? TNF driven pathway of necroptosis through RIPK1, RIPK3 is very important in innate immune control of the viral infection. Now, it turns out that in recent years, the last 2-3 years, a second pathway that inhibits necroptosis has also been discovered in pox virus. So this is work done by Ed McCoskey and Bertrand Jacobs at Arizona State. And so this is a graphical summary and taken out from one of their diagrams. So to cut a long story short, so it turns out that in the pox virus genome, they encode a viral protein called E3L. So this E3L molecule, is a very interesting molecule. It has 2 parts, and one of the parts turns out is encodes a domain called the C alpha domain, and we're going to talk a little more about that later on. And this C alpha domain is a portion that can actually bind to nucleic acid. And so one of the other RIM containing adapter that I told you earlier is CBP1. It turns out CBP1 also has a C alpha domain. So you can imagine that the C alpha domain being a nucleic acid binding domain, the virus encoding one, and of course when the virus infects a cell, is of course actively transcribing is sort of like genes, and so there's nucleic acid, viral nucleic acid generated. And so maybe, and of course, we know nucleic acid, RNA, whatnot, they are strong inducer of innate inflammation as well. So you can imagine that E3L, the viral protein, through its C alpha domain, its function is to sequester these virally generated RNA and therefore prevent CB1 activation. And so what Bertrand Jacob they were able to show is that if you inactivate or remove this C alpha domain from this E3L viral protein, now the virus can no longer sequester these viral nucleic acid, and the nucleic acid will then go on and bind to CBP1 and subsequently activate RIPK3 and necroptosis. Now, I need to emphasize that this pathway is a separate necroptosis inhibitory pathway, separate from the TNF pathway that I referred to a moment ago. So there are two, in the pox virus, there are two separate necroptosis inhibitory pathway. One mediated by VRRD and the second one mediated by E3L. Now it turns out that pox viruses don't just infect in mammals, so you also have pox viruses. They're more distant family members. So AV pox viruses, for example, they infect birds. Okay, and so it turns out that James Murphy at WeHi, he several years ago found a class of viral MLKL homologs that he showed when overexpressed in mammalian cells can actually compete with and inhibit necroptosis by competing with the mammalian MLKL for binding to RIPK3. However, it is important to emphasize that in addition to what he found in the AVPOX virus, viral MLKL orthologs were also found in myxoma virus. Now, you'll remember what I told you a moment ago, right, with myxoma virus. And you'll also remember that, in our experiment, the experiment I showed you, earlier, that myxoma virus infected cells, despite, the virus encoding a viral MLKL, the infected cells are still sensitive to necroptosis. So it looks like, that perhaps in this case, this viral MLKL inhibitor is only active, in other species, but they are not relevant, for mammalian pox viruses. So this is something that I just want to emphasize. So we spent a great deal of time to talk about pox viruses, but I want to switch gear now and talk a little bit about herpes viruses. It turns out that herpes viruses, although they're also large DNA virus that can inhibit caspases, they use a different strategy to block necroptosis. And so the work I'm going to tell you about, a lot of it was done by Ed McCaskey. And so here is a paper that they published quite early on, more like than 10 years ago now, where they discover a viral protein encoded by the mouse cytomegalovirus, or MVMV. So this virus encodes a ribonucleotide reductase-like protein as a pretty large protein. Now, although this protein, the structure, has a domain that looks like a ribonucleotide reductase, but this ribonucleotide reductase domain actually has no enzymatic activity. What it does have is at the end terminus a rim motif. So you will remember, I told you earlier, the rim is the protein interaction motif that nucleate and allow the necroptosis adapters to come together. So it is essential. So it is very interesting, that the virus actually encodes a protein that also has a rim domain. So is it actually important for viral pathogenesis? So what Ed McCaskey, their group did was to create a mutant virus that has a mutation that knocks out the function of the rim motif in the M45 viral protein here. And so when you did that, you can see, compared with wild-type virus infection. So in wild-type virus, the virus, after a couple of days, they replicate and reach a peak, after several days of infection, and eventually the immune response kicks in and control the viral replication. So this is the sort of like classic kinetics that you observe with foot pad infection in MCMV infection. Now, if you, however, were to inactivate this viral protein RIM motif, then and perform infection in the mouse. You can see that now that this virus can no longer induce a productive infection in the mouse. And the reason is because now the virus cannot inhibit necroptosis, and you have basically spontaneous and premature activation of cell death, and that prevents replication of the virus. So what is the RIM, right? Just want to briefly mention. So the RIM, is basically an acronym that stands for RIP, homotypic interaction motif. It is actually first defined by Vishwadixit, all the way back in 2002. So in recent times, we and other people have shown that the RIM motif actually allows these adapters to form an amyloid-like structures shown here in the EM and also by fluorescence microscopy. And this amyloid formation is absolutely essential for promotion of necroptosis. So now I told you already that the necroptosis induced in the absence of this viral inhibitor is going through RIPK3, but then what is the upstream activator? So we know in the case of pox virus, I told you earlier that TNF is a major inducer of necroptosis through RIPK1 and RIPK3. What about the MCMV here? So it turns out in MCMV RIPK1 is not essential, but instead it is CBP1 that is actually responsible, and the mechanism for CBP1 you can see once again. So if you again using the same mutant MCMV virus that lacks a functional RIM motif, they cannot have a productive infection. But then if you were to do the infection now in an animal that lacks CBP1, then you can see now you can restore normal infection kinetics even with the mutant virus that do not have the functional RIM motif. So this process once again is through sensing of viral RNA by the CBP1 adapter. So so that's MCMV is a mouse cytomegalovirus, right? So what about human herpes virus? So it turns out that if you go to human viruses, right? Here we're looking at human simplex virus 1 and 2. They also encode a M45-like molecule. In the case of HSV1, is a molecule by the name of ICP6. In HSV2 is ICP10. Both of them has a very similar architecture, a ribonucleotide reductase-like motif, and also RIM at the N-terminus. And the situation is very similar. So the RIM in the ICP molecule, they are responsible for inhibition of necroptosis. And when you introduce mutations or you knock out this viral gene, then the virus-infected cells become sensitized to necroptosis. So now, HSV1 and HSV2, these are human pathogens, right? So what is quite interesting is the ICP6 and ICP10 molecules in human cell, they function as inhibitors of necroptosis. But if we were to actually do the same experiment now in mouse cells, surprisingly enough, it actually causes apoptosis. So shown in these graphs here, this is malinfection. So remember, it is a human pathogen. You can, however, use a lot of this virus and to establish a productive infection in mice, but then it is not physiological. And what is quite striking is, in this case, you can look at all these phenotypes here, and you can see that the inhibitor is actually a driver of necroptosis. So what I like to argue, and I think this is very clear evidence to indicate that maybe these cell death adapters, beyond just controlling host cell death during a productive infection, may also confer species restriction. We know that many viruses, right, they only infect certain species, and thank goodness, right, they don't often jump from species to species. And maybe some of these molecules are responsible for this species restriction. So this is just the cartoon to illustrate what I just mentioned to you a moment ago. Mouse infection, human infection, you can have the same inhibitor, but then they can have completely different outcome. So and this just for completeness sake, just to mention that varicella zoster is a virus that causes shingle, and it also is a herpes virus, and it also has a rim-like inhibitor that behave very similar to the M45 and the ICP that I mentioned to you a moment ago. Now, however, what is interesting, and it is an interesting exception, is human cytomegalovirus. So again, this we're still talking about herpes virus. So in human cytomegalovirus, right, the virus infection also causes inhibition of necroptosis. So that's been known for a number of years. But then, funnily enough, however, we were not able to find a M45-like molecule that encode a rim motif that inhibits necroptosis. So it indicates that there's some other distinct mechanisms responsible for inhibition of necroptosis with this human cytomegalovirus. And so recently, it was discovered that in human CMV, and of course, many of us actually carry CMV, right? They encode this molecule called UL36, and this inhibitor has a dual function. It inhibits caspase A, but at the same time, it can also bind to MLKL to induce its proteasome degradation. So it's one molecule here that can inhibit both apoptosis and necroptosis. Now I know that the topic for today in the discussion focuses on viral pathogen, but I can't help to just mention it in passing. That it turns out that it's not just viruses that care enough about necroptosis to encode mechanisms to block the cell death pathway, but then certain pathogenic bacteria also do the same thing. So here is a study that I'm citing here from Elizabeth Hartland in Australia, where a few years ago they discovered that in certain enteric bacteria that causes diarrhea, for example, they encode a class of atypical citizen proteases called ESPL that can cleave and inactivate rim-containing adapters. So in this case, however, it is not specific against just RIPK3. It can also cleave and inactivate other rim adapters, including RIPK1, TRIF, and also CBP1. So what we're looking at basically is for pathogens, right? Viruses in particular, they encode many different types of strategies to inhibit necroptosis. I mentioned today about VRD from pox viruses. It causes degradation of RIPK3 through the proteasome. And then with herpes viruses, right? In many cases, they encode mechanisms and inhibitors that inhibit necroptosis through a rim-mediated sequestration of the essential necroptosis adapters in the host. And of course, there's the bacterial pathogens that I mentioned just now that can cleave and inactivate the rim adapters. So all of that, I think, a pretty strong argument, right? To argue that why do we have necroptosis? Why do we have apoptosis and all these different types of cell death programs? I think when you have so many pathogens, development strategies, and they care enough about the pathway to find ways to actually block it, I think it's a pretty strong argument that necroptosis perhaps is a evolutionary product that we develop, to sort of deal with infection by pathogens. However, right, what I show you, so far, right, all the examples I show you, most of them, they were kind of in mouse infection recently, right, so if that's so important, right, do we see real examples, right, in human infection? And so recently, and this is really, just this year, early this year, right, Lauren Casanova in the group, they found a patient. So this is the family tree and the pedigree, for the patient here, the P1 patient. So this patient, get a sort of mutated allele, of RIPK3 from both parents, Okay, the alleles, the mutant alleles, shown here, in both cases, causing, either, a premature, frame shift, termination of the RIPK3 protein, or a point mutation. So in both cases, it causes basically, instability of RIPK3 protein. Of course, in this case, for this allele, it completely, deletes, the rim motif. And funnily enough, right, this patient was identified because it suffered, from childhood, developed, herpes simplex, virus, induced, encephalitis. And so you will remember, what I just told you a moment ago, right? In mouse, herpes virus infection, for example, if you can have no RIPK3, but normal virus infection, there's no phenotype whatsoever. But in this case, in human virus infection, if you do not have RIPK3, at least from this one single patient, right? Of course, more work needs to be done to really demonstrate whether this is really causative or not. But it is nonetheless interesting to raise the possibility that perhaps what we found in mice, right? We have to take it, with a grain of caution, because in human, if you do not have RIPK3, there's the possibility that we can't really, sufficiently deal with, even a pathogen that otherwise encode an inhibitor of necroptosis. So we've talked about, pox virus. We've talked about, herpes virus. These are both, large DNA viruses. They are well known to encode, immune modulatory genes, as I mentioned, at the very beginning. What about RNA viruses? So we're going to touch upon, just briefly in the last couple of minutes, certain RNA viruses. So the most well-known example is influenza virus. So it turns out that many studies that I'm listing here, right, have demonstrated over the last couple of years that influenza virus through a CPP1, CPP1 has another name called DAI, can be activated, and then go on to trigger, RIPK3 activation and necroptosis, or in some cases apoptosis as well, as a way, to sort of deal with this infection. So to look at, some of the results, taken out from these studies, you can see, for example, in tissue culture infection, right? If you were to take out RIPK3, you can see, now that you get reduced cell death induced by influenza A, virus infection. And then if you look at, mouse infection without, RIPK3, this lack of cell death actually is causing, also, sort of a compromise, survival, for the animal. So virus-induced cell death, through RIPK3 is important for controlling, influenza A, virus infection. Now, however, that was that. If you go on now and look at some of the RNA virus infection, we get a slightly different picture here. So here's an exact, a report that I'm citing here is a study, done by Andrew Obers at University of Washington, where he looked at West Nile virus. So it's just a neurotropic, flavivirus, and look at whether the RIPK3 is actually important. You can see, in the survival curve, here in red, animal without RIPK3, they succumb to the infection. So again, showing that RIPK3 is important. Now you might say that, oh wow, okay. So what, most likely, it's just like, what I show you throughout the last hour, that, okay, RIPK3 induced, necroptosis or cell death is important for this, protective effect. But it turns out that, that to be not the case, because you can actually look at animal that doesn't have MLKL. Remember, MLKL is a downstream effector molecule for RIPK3. So if this effect, protective effect, driven by RIPK3 is through necroptosis, you should see the same, deficiency and mortality in the MLKL knockout. It turns out that we don't see that. You don't have MLKL, you get the same response. So again, to tell a long story short, right, what Andrew, were able to show is that in this particular case, for this neurotropic virus infection, RIPK3 is actually conferring protection not by cell death, but through production of chemokines. to draw basically immune cells into the infected site to control and contain the virus infection. And that turned out to be, not the only case, for West Nile virus, but it goes beyond that, and in this, follow-up study, also done by Andrew, he looked at Zika virus. So again, it's a, flavivirus. Zika virus, of course, like for you all, in Brazil, you will need no introduction, with this, virus. And so in this study, looking at, flavivirus, it was actually quite surprising because once again, it is independent of the ability of RIPK3 to drive cell death. But what he was able to show is RIPK3 through a pathway that promote IRF1 induced gene expression and production of a metabolite, itokinate, which we know now has many immune modulatory function. So this is how RIPK3 actually confer protection against this pathogen infection. So it looks like RIPK3 can have sort of like cell death independent activity in virus infections as well. Okay. And so this, in fact, is actually no surprise because we have actually seen this type of crosstalk before, way back, about 10 years ago. So there's a study, done by James Vince at WeHi, where he showed that under certain conditions, RIPK3, beyond just promoting cell death, can actually promote not-free inflammasome maturation to promote IL-1 beta production. We also later on have seen that in bone marrow-derived dendritic cells. RIPK3 is essential for also production of IL-1 beta if you just tickle them with LPS in the absence of an inflammasome agonist. So now, that's RIPK3, but then MOKL, going beyond the RIPK3. Now we also appreciate that, when the cells, undergo necroptosis and therefore activating MOKL can also indirectly activate and crosstalk, with the inflammasome pathway, because once you have the pores formed through MOKL organization and membrane targeting, then you can have ions leaking out from the cell, and one of these ions could be potassium. So we know potassium efflux is a key priming signal for inflammasome activation, and so that can therefore indirectly be activating the inflammasome that way. So RIPK3, MOKL can separately and independently promote inflammasome activation through distinct mechanisms. And then, of course, this relationship, goes the other way as well, right? So recently, there's also been some studies implicating, possible, sort of like crosstalk, from inflammasome activation to also promoting necroptosis. So in this study, the authors are looking at basically a possible role, for this gene RIPK2. It's a Parkinson's disease-associated gene. It's also involving and associated with Crohn's disease and also for susceptibility to mycobacterial infection. So in this study, the authors look at just basically mycobacterial infection, and what they found was in this mycobacterial infection, you can get inflammasome activation, and that's not surprising. But then this inflammatory activation beyond doing what it normally does, for example, puncturing membrane, it can also actually alter the mitochondrial membrane permeability. In doing so, generating reactive oxygen species that can actually then go on and activate and promote RIPK1 and RIPK3 activation and necroptosis. And of course, finally, I want to wrap it up, by saying that, I think, the word permaptosis, if you read, recently, it's an idea, that is being pioneered, by Turu Kaneganti, and so she's been, basically, pushing this idea that influenza virus, infection in particular, but then she's now, go beyond that and showing that, in other situations, possibly, that through CP1 activation, one can actually drive formation of this macromolecular complex that contains all these adaptive molecules, including RIPK3, caspase A, NAL3, so on and so forth. So all these components, of course, they regulate and drive different types of cell death and also inflammasome activation. So she used the term panoptosis to refer to the possibility that in certain situations, one can actually drive multiple different types of cell death programs at the same time in the same cell. But I think this still will need further experimental validation. Okay, so I want to kind of close by showing this little cartoon that I take out from the CDC in the US. So we started this conversation focusing on the discussion of pox virus and how pox viruses interact with the host necroptosis machinery. What is interesting to remember is pox virus is actually a pretty ancient virus, right? It's only because of the success of the vaccination program worldwide that we no longer have to worry about pox virus or smallpox as a human pathogen. But if you go back to ancient Chinese history and as early as you have written history, right? Ancient Chinese history or Egyptian written history, you can see records of describing basically smallpox like disease. So that means it is a pathogen that has been with us for a long time. And it is also a pathogen that appears to care a great deal about this necroptosis machinery in encoding not only one, but as I showed today, a second pathway that interfere with this necroptosis response. So I think this types of sort of like anecdotal evidence and observations make a strong argument that indeed I think necroptosis has a pretty important role in this pathogen and host or host pathogen coevolution sort of like journey. So with that, I think I'll stop and maybe I'll just see if there are any questions from people.
Francis, absolutely sensational.
My gosh, Francis.
Really.
What a lecture. My gosh, stunning.
Brilliant.
I hope it's clear.
This is brilliant. I've been hearing, lectures on necroptosis, but yours were just
my gosh, incredible.
Everything is concatenated with previous information, so it's really cool. Really, thank you so much.
What a linear thoughts. Every question I had, you answer the following slide. My gosh, it was fantastic.
All right,
I
so
love it.
Let's start questioning by students. Abology.
Okay. Yes. Fantastic. Great lecture. It was really, really cool. But I got some new information, which I would just like to ask a few questions about and just to verify. So the first one, I think I have close to 5 or 6 questions here. So the first one is number one. From the lecture, you said that the TNF receptor, once it's activated, can activate the RIPK1, then RIPK1, phosphorylate RIPK3, then downstream MK, then necroptosis. Then at some point also you mentioned about TRIF, about toll-like receptor 3 and 4, and right. Can directly activate RIPK3, then necroptosis. Also for the ZBP1, which is for virus. So my question is, does it mean that necroptosis or RIPK3 activation depends specifically on the receptor or the pathogen?
Well, I think, it depends on the context, right? So toll-like receptor 3 and 4, for example, right? So these are sort of like, the PRRs, and they're mostly, for example, like toll-like receptor 4, there'll be like, sort of like, LPS sensor, right? So maybe it will have a stronger role, in certain types of bacterial infection. And towards the end, I mentioned mycobacteria infections, right? So that can be one situation where, like, the total receptor and then TRIF, and the RIPK3, pathway can be engaged. So for CPP1, I gave you several examples. So RNA viruses, for example, or even DNA viruses, right? Except that in the case of DNA viruses, mostly in a normal situation for a wild type virus, right? They usually, have ways to actually block, the CP1 activation. But for RNA virus such as influenza, then you get, the virus, because it's a very simple virus, right? It only encodes several genes. So they don't really have ways to interfere with this CBP1 activation. So because of that, you see CBP1 activation, and then downstream effector molecule also engage, such as RIPK3 and MLKL.
Okay.
So it's context dependent.
Okay, thank you. Then also another question is that you mentioned that RIPK3 helps to control infection, and in RIPK3 deficient mice, that the mice actually die of infection, So my question is this applicable in all conditions, irrespective of the pathogen, that virus or bacteria, RIPK3 can help to control this infection?
Right, so it is not, so I need to emphasize, right, so it is only required for certain types of pathogen. So we've done like, beyond, what I, told you, today, and other people have done it too, right? So you can take, for example, LCMV, okay? And, you don't have to have RIPK3, the mice do perfectly fine. In fact, you can do, like, a lot, even like, for some of the, pox viruses, right? They are okay, I mean, like there's some effects, on, viral replication, but ultimately, if you don't use sort of like a lethal dose, they find a way, to sort of like overcome it. But, like there are certainly, examples where, you need, this pathway, to control and contain the pathogen. And I think, again, I gave you examples, today, like, what those situations are. But certainly, you can look at, many types of, viral pathogens, and they don't really seem to actually care much about the pathway. So it's very specific, right? And I think, again, I make the argument that, so for example, so there are like, of course, COVID, like, SARS, COVID-2, COVID-19. So there's been studies, you can easily do a PubMed, search, lots of papers, saying that, okay, well, SARS-CoV-2 can induce, different types of cell death. I believe it, okay, but it is a very recent pathogen, and I think it's inducing a lot of like, different cell death programs, in part because in any infection, for that matter, you have lots of cytokines produced. So we talked about, TNF today, right? So TNF can induce cell death. We talk about interferon. So interferon, among other things, can also turn on, for example, CBP1 expression. So that, you can turn on another way that can sensitize cells to necroptosis, right? So it is always a myriad of reaction and responses that the host is actually engaging in response to any certain pathogen. But it is a very short history, right? So what I like to think, is when we actually sort of like, stepped aside, from our vantage point, right, and like, step into the shoes of the virus, right, and think like a virus. What do I care the most about? And I think, when you do that, you look at, these like viruses, for example, that has very defined and specific machinery to target, this pathway. I think it speaks volumes about, why, this pathway is important. Yeah, so that's kind of like, how I think about, the situation here.
Okay, all right, thank you. Then also you talked about how cowpox involved in the disappearance of RIPK3 protein expression. I don't understand how does this happen. And two is, do you think it would be ideal, because in our lab here we use Listeria, we use adenovirus. Do you think it would be ideal to investigate if this Listeria adenovirus could also cause maybe RIPK3 protein, disappearance of RIPK3 protein expression?
So this is, which virus are you talking about again? I'm sorry, I missed that.
You talked about cowpox, but
Right.
then I'm giving you an example of adenovirus or Listeria.
Adenovirus. Okay, yeah. Adenovirus, no. So we have not seen evidence, no, of adenovirus encoding anything that can interfere with necroptosis. But I have to confess, that I have not looked very extensively, in the area. But we looked, we did, okay. But we did not, I guess, the early returns, we don't really see any evidence, and therefore we don't follow on that. Is it possible? I mean, could be possible, that we missed something, but I don't really see that. And also, when you look at the viral genome, all the viral proteins, that you can see, they don't really have any features, that makes you sort of like think that, oh, maybe, some of these, viral proteins are interfering with necroptosis. So, because of that, I don't think it is, but I cannot say definitively because we have looked, but not extensively.
Okay. My next question. Also, RIPK3, that you said it helps to control infection. So, do you think it will be important in the induction of anti-tumor immune response also?
Yeah, so that actually is a subject, of intense interest, for a lot of people, right? So immunogenic cell death, I mean, Guido Kramer, like, Peter Van den Billy, Dimitri Crisco, Andrew Obers, lots of these people, they have used different strategies and models to show that you can induce necroptosis as a way to induce immunogenic cell death and therefore boost your anti-tumor response, right? And we have also began to do something in that direction. So I agree, with all those people's, right, their studies, what they concluded, and that is that yes, it is possible to induce an anti-tumor inflammatory response as a way to boost anti-tumor immunity. But it is just like, for example, like Fanshaw, has papers looking at parotosis, right, as a way to induce immunogenic cell death and also anti-tumor immunity. And then ferroptosis, of course, is the rage of the time now, right? There's so much on ferroptosis and cancer. So I think all of these, of course, are immunogenic cell death, right? They're lytic cell death, and so at that level, you can say that they're similar. But I think when you think about all these different ways to do it, I think experimentally we can demonstrate it. Going from experimental evidence to actually, like, making it work in the clinic, I think there's still a long way to go. I always like to think that in immunogenic cell death is just really one component. It is important, but the duration and magnitude of the signal that you provide is equally important. Too much of a good thing is bad, right? So when you have, like, and it's just like biology, right? So I think, with immunogenic cell death, it's a similar situation. How long do you provide, that stimulus? And then the magnitude. These all matters. So in our experience as well, we see the same thing. The magnitude and the duration is important.
Okay, so thank you. Lastly,
Your apology. Let me move to another people's questions. Save yours, and I'll come back to you. Okay.
right.
So we don't monopolize Francis with your questions. Pamela, please.
Hi, Professor. Thank you for your presentation. And I had some doubts with panoptosis. This occurs in other microorganisms.
Are we still being recorded?
Why?
Are we still being recorded?
Yeah, you want, we can. Okay.
I have to be careful, okay?
We can stop the recording now if you, I think we can do that.
That's perfect, yeah.
Márcio, I don't know if you hearing us.
Tô aqui ouvindo.
Então, se você puder terminar, so the lecture is over. We thank people from the YouTube, and now we move only to this room. Você pode parar, então,